Welcome to Exam Mini Video 2. Today we are going to be talking about epistasis, specifically the single dominant mode of epistasis. Again, here are the basic terms you should know to understand this topic. Please pause and review if need be. So what is epistasis? Epistasis is a phenomenon in which the expression of alleles at one locus masks or modifies the expression of other alleles at another locus, so two genes are coding for the same trait. Note that a locus is a specific physical location on a chromosome where a gene is located. There are many modes of epistasis, but today we will focus on the single dominant epistasis, a mode in which a dominant allele A masks or modifies the expression of alleles at a second locus B. The A allele is epistatic to B, meaning that any genotype with a dominant A allele will produce the same phenotype regardless of what alleles are at the B locus. We can see that in genotypes with the dominant A allele, the A activates an inhibitor, represented by the T-bar, which represses the production of enzyme 1, preventing the precursor phenotype from converting to the intermediate phenotype, expressing the precursor phenotype. In the genotype that does not have a dominant A or B allele, there is no epistatic inhibition of enzyme 1 via the dominant A allele, so enzyme 1 is produced. But there is not a dominant B allele to produce enzyme 2, so the precursor phenotype is able to convert to the intermediate phenotype, but not progress to the end product phenotype, expressing the intermediate phenotype. In genotypes that do not have a dominant A allele but have a dominant B allele, there is no epistatic inhibition of enzyme 1, so enzyme 1 is produced, and there is a dominant B allele that produces enzyme 2, which allows the resulting intermediate phenotype to convert to the end product phenotype. We can think of this epistatic pathway like a railroad in which the train is the genotype, the genotrain if you will, and the cars are the loci holding their respective alleles. As the train moves along the track, it encounters railway junctions at the enzyme arrows and inhibition T-bar. The alleles control these junctions, switching the track to guide the train to its expressed phenotype. In this example, the precursor is a blue elephant, the intermediate a purple elephant, and the end product a pink elephant. Let's see what elephant each genotrain picks up. As seen before, genotypes containing a dominant epistatic A will produce the same phenotype, no matter what is happening at the B locus. These genotypes repress the production of enzyme 1, switching the track to guide the genotrain to the blue elephant. Genotypes containing no dominant A allele and no dominant B allele allow the production of enzyme 1 but not enzyme 2, switching the track to guide the genotrain to the purple elephant. Genotypes containing no dominant A allele that do have a dominant B allele allow the production of both enzyme 1 and 2, switching the track to guide the genotrain to the pink elephant. What would be the progeny ratio if you cross parents who were both heterozygous for the A and B alleles? We can see in the dihybrid Punnett square that all genotypes containing a dominant A allele express the blue precursor phenotype due to the epistatic repression of enzyme 1. All genotypes without a dominant A or dominant B allele express the intermediate purple phenotype because of the production of enzyme 1 but not enzyme 2. All genotypes without a dominant A allele but with a dominant B allele express the end product pink phenotype because of the production of both enzyme 1 and 2. If we count and sort the squares according to their phenotypes, we can see that the progeny ratio of this cross is 9 to 3 to 1. Let's look at one more example and cross one parent who is heterozygous A and homozygous recessive B and another parent who is homozygous recessive A and heterozygous B. The result looks a bit different than the last cross, but the same principles apply. By counting and sorting according to the phenotypes again, we see that the progeny ratio of this cross is now 8 to 4 to 4. Here are my references. That is the end of this video. Stay tuned for another in 2 weeks.